In order to get a good understanding of how carcinogenesis works in the formation of malignant neoplasms, we need to understand basics in two pretty complex cellular uh, life cycles, right? So, differentiation and cellular adaptation. They're complex. I'll try and go slow and easy here. So, we'll start with differentiation. So, when a cell is born, let's start with uh, yeah, let's start with a stem cell, a multi-potential hematopoietic stem cell. Hematopoietic stem cell. So, baby in the bone marrow. And through the process of differentiation, this stem cell may go into it can go into a myeloid progenitor cell or a lymphoid progenitor cell, and then as it goes from there, it may go into it can go into red blood cells, it can go into platelets, it could go into megakaryocytes, it could go to you know, actually all sorts of different white blood cells except for lymphocytes. And then this side, you get the lymphocytes over here that go into different B and T cells. What's going on is as this cell is born, it goes through a process of differentiation where it goes and it becomes different as it goes through its life stages, and it becomes what it needs to be when it grows up, if you want to call it. So through the process of differentiated, a mature red blood cell, a mature thrombocyte, a mature B type lymphocyte, those are all fully differentiated cells. So these are cells that are fully functioning, that are intending to do what they should be doing. I love spelling while I'm talking. All right, so where does it all go wrong, right? So as we're getting towards carcinogenesis, we have to take a look at the different phases of cellular adaptation. So a normal cell will look like this, right under the microscope, nice and aligned. The structure is very nice and neat. They're following what's called contact inhibition, meaning that they're sitting side by side, not on top of each other, not disorganized. They're nice and aligned as they should be. And this is normal, right? There's a couple of different ways that this could go. These cells, if they don't get the nutrients they need, they aren't activated with physical activity, they may shrink and atrophy, which is the shrinking of cells. They don't become less numerous, they just physically become smaller. Or if someone was to work out those muscles in the gym for a long period of time, or if some sort of hormonal cause caused cells to grow larger, this would be called hypertrophy or enlarging. So this could be disease or it could be muscle building. Where it starts to go from these types to more down into being less differentiated, not so useful, is if they become hyperplastic or hyperplasia. So, excuse me, I spelled that wrong. Hyperplasia. So hyperplasia is when these cells become more numerous. They're not growing on top of each other. They're crowding each other. They're getting really, really tight on top of each other. A really good example of that is if a virus, HPV, gets inside of epithelial cells, those cells will become so numerous that they start packing on top of each other and they start pushing up to the top in a wart. That's a normal skin wart seen in human papillomavirus. Or it might be a hormonal imbalance may cause extra tissue, extra cells to form in the prostate, causing prostate to squeeze around the urinary tract and the urethra, causing difficulty urinating in older men. Okay, moving along. So these are definitely not signs of cancer just yet. These may be completely unrelated to cancer, especially these, of course. This on our way here, this is not necessarily a sign of cancer for sure. But as we continue to go further, we get into dysplasia, and this is where it starts to go bad. So bad growth, right? Excessive growth, right? So Dysplasia is now these cells they alternate size, shape, and organization. So some of them are really large, some of them are small. They're growing on top of each other. They're growing way disorganized. This is bad. This is potentially a precursor for a malignant neoplasm formation. And then as it continues along, there may be a stage of metaplasia, which is a change in growth. So what this might happen to is that certain types of cells may turn into totally different types of cells. A very good example is in the airway, the columnar epithelial cells that line the airway, they are changed to different cells as the cilia are destroyed over long-term exposure of smoke, and those cells are actually changed, and that's another form. It doesn't have to pass through this. It may go directly from dysplasia to what's called neoplasia. And neoplasia is new type of cell or new growth, and those are cells that they don't look anything like those parent cells. And when they get to this point, they are called what's fully undifferentiated. Fully undifferentiated, these neoplastic cells are. They are undifferentiated because they are not fully functioning. These do nothing positive for the body. They serve no purpose. They will not do anything positive. They will only, at this point, this may be a cancer cell or it may not be, but it is called a neoplasm. And a neoplasm can be benign or it can be malignant. But either way, these are called undifferentiated. But it's qualities of how they grow that makes it cancerous, being a malignant neoplasm or not. In this study, we'll be focusing more on carcinogenesis, or the production of how it transforms from neoplasia into a malignant neoplasm, and where does it go from there.